any language, programming language you desire, you'll be implementing the binary programming solution, or you're not going to be implementing the original one. I'm going to modify, make some modifications so that you have to implement this new algorithm, because you cannot. There's lots of code on the internet. You can find global alignments, local alignments, semi-global alignments, source code directly. So I'm going to have you implement a certain the alignment for a new problem. Okay? So you will have to actually implement. You will not be able to find the source code elsewhere, but you can make use of existing source code. If you're allowed to use any internet sources in your homework, but you should know the binary programming algorithm, and you'll be able to. You'll be implementing. Okay. So if you have any questions about the local alignment solution or the binary programming algorithm, how do you fill the tables, you can ask now. Otherwise, I'm going to assume that everything is crystal clear. Okay. Does anyone have any questions? Okay, so let's go ahead and talk about database searches. As I said, I'm not going to go into details, implementation details of BLAST and FASTA. This is just going to give you some idea about how these database searches work and how they work fast. The BLAST is the acronym for Basic Local Alignment Search Tool. I told you that it's a very popular algorithm. It's made into the English dictionary as a verb. And it's a heuristic method for local alignment. Heuristic means that it's based on a certain intuitive idea to do the searching, but it does not guarantee you to find the optimal solution. And it's designed mainly specifically with this goal in mind. Okay, we have a query sequence. We are going to have millions of sequences like this, and what we want to do is we want to do a search like Google search. Okay, imagine this is documents on the web. What you do in Google search is you enter some keywords, and you want to get the results real fast. Okay, this is the main issue: efficiency, speed efficiency. If you cannot get your results fast, it has to be interactive, it has to be really fast, because you'll be probably conducting many different searches for many sequences. So here the main goal is to do this search efficiently. And exhaustive search is our option. Okay, you cannot think to do this, accomplish this by making this comparison one by one to all the database versions. So, in the area of database research, okay, database research, there is this term called indexing. What we need to do, I mean, you probably know what indexing means, and Google also, Google Desktop has this. What it does is it indexes your local documents on your PC so that you can conduct your searches faster. So what does this indexing mean? For example, a telephone book should be indexed somehow. It's just because in order to find who you are looking for in a phone book, you do not go over all the names in the book, right? It's going to take days. So indexing is a way to organize your database, your original data. You organize your data in such a way that the searches can be made fast. There are many different techniques for indexing in especially multi-dimensional databases. And here we also have a multi-dimensional data here. Our data here, our individual data here, is a protein or DNA sequence. And this protein or DNA sequence is multi-dimensional because it's composed of an array of characters. Okay? Sometimes 400-dimensional, sometimes 1,000-dimensional. It's very unique that we do not have fixed length DNA or protein sequences. Now, one way, a very simple way to do this indexing is if you are looking for a certain sequence by their first amino acids, you could organize them as in a dictionary, right? you could basically sort them in lexicographic order, meaning that put the sequences which begin with A at the beginning, put the sequences which begin with C at the end, and T and G and T, for example, comes later, and sort the remaining according to the rest, the second nucleotide letters. But is this meaningful? What is it going to give you? I mean, we are not searching for sequences by their beginning letters. It's not like we are searching for someone in a phone book when you do this search. Okay? What we are doing is we are trying to find similar sequences to this one. So lexicographic ordering does not do us any good, because our sequence may have a certain similar region to database sequence, which is somewhere in the middle of the sequence. It may not be at the beginning. You cannot search your sequence as if you're searching a phone book. So some more intelligent and different indexing strategies should be devised. Now, one idea is this. When we are comparing these, when we have this database, if we consider it as a fixed, not dynamic, not very frequently updated database, what we can do is, which is going to take a very long time actually, the solution I'm going to tell you right now, is to somehow, this database, if you think about it as a sequence space, in this database, some of the sequences are going to be more similar to each other, right? And even in this 10 million sequences, they are not random sequences. Some of these 10 million sequences will be similar to each other, up higher than a certain threshold. Some other, for example, 1 million of these sequences are similar to each other more than this 40% sequence identity. And maybe another 1 million, they're also similar to each other. These are probably these are certainly not correct figures, correct numbers, but I'm just giving you an idea that these 10 billion sequences in our database, they have similarities with respect to each other, not considering our query sequence. So what we can, a very common way to do indexing is to cluster your database hierarchically by using these different similarity levels. So what is it going to do? What we can do is, instead of comparing our sequence to these 10 million sequences one by one, we can compare our sequence to one representative, to one sequence from each of these 4 clusters. So if we do that, what we can have an idea of our sequence is most similar to one of these 4 clusters. If we can get to that, what we can do is we can immediately, for example, we have found that out that our query is very similar to the sequence, a representative sequence here. Okay, by just doing four alignments, we can find out which one of these 4 clusters our sequence is closest to. And then, if we find that our query sequence actually is similar to a region here, we can immediately forget about making alignment for these 3 clusters. So it allowed us to prune out 75% of the database. Okay? So this, BLAST has nothing to do with this. Okay? This idea is just a general idea that is used in database indexing. What we did was, we organized our data. So this is just an example of data organization. We organized our data in a certain way, which allowed us, during the searching, we were able to speed up our search a lot more by discarding most of the database when we are doing the search. Okay? So we need strategies like this. If we want to speed up the search, we need strategies like this. So what BLAST uses a different strategy based on this observation. This observation is good alignments contain short lengths of exact matches. So what is it going to help us? We are going to see. The idea is like this. doesn't use this, BLAST does not use this clustering idea. But again, it pre-processes the database so that, so one thing in indexing is that we need to pre-process these 10 million sequences. We need to run some algorithm, run some indexing algorithm on these 10 million sequences and convert it to some other representation which is going to allow us to do the search fast. Okay? BLAST cannot run directly on 10 million FASTA files. Okay? FASTA is the sequence file format. Okay? Or 10 million sequences. If you write your sequences one after another in a text file, okay, 10 million sequences in a text file, and you say, okay, I want to do BLAST search on this 10 million sequences, which are put there one after another, one by one. The thing is, the BLAST algorithm is not only doing the search, indexing is part of the BLAST algorithm as well. If you say I want to run BLAST on these 10 million sequence files or 10 million sequence entries in a file, BLAST won't run. Because what BLAST needs is, BLAST needs these sequences to be processed and indexed so that can be in a search search faster. Indexing is going to convert these 10 million sequences into a format that can be searched faster. And what is this format for? So therefore, BLAST has these two components. When you're building a database, you need to index it, and meaning that you process it, you convert it to some other format. In some cases, these indexes can be much larger than the original sequence file itself. Okay, so this is also another factor in database research. This database research area is, okay, you generate an index file for your sequences. What is the size of your index file? If the generated index, for example, if the generated index is on the order of n squared, for example, if you have n sequences, okay, this is going to be unacceptable. Okay, it means that, for example, if you need 1 terabyte to store all your 10 million sequences, for your index, you are going to need 1 terabyte times 1 terabyte storage. And that's how many billion? So it's not billion, right? It's terabyte. I mean, giga is billion. So it's 1,000 billion terabytes. You are going to need that much storage to store your index. So the index you generate should also be in comparable size to your database. So this is what BLAST does to do the indexing. For each database sequence, BLAST processes them separately. And for DNA sequences, for example, it slides an 11 base pair length window over the sequence, and it basically has a hash table of these length 11 words. So now let's think about this. If we are only considering length 11 words, okay? So how many different words can we have? It's 4 to the 11, right? If imagine you have a sequence of length 11. If you are dealing with DNA sequences, how many different such words can you have in your dictionary? It's 4 to the 11. Okay? So it's a manageable number. 4 to the 11 is like 2 to the 22, right? So it's like this. So it's 2 to the 22. So what is it? It's like 1 million? Something like that, right? No, it's 1 million. No, it's 1 billion. 2 to the 10 is like 1 million. No, it's 1,000. 2 to the 10 is 1,000. So it's about 1 million, something like that. It's around millions, right? It's around maybe 4 million. Yes, 4 or 5 million or maybe 8 million. But it's at the order of millions. So the thing is, a million is not I mean, it's still your index. If you have 10 million sequences in your entry, in your index, you are going to have again entries on the order of millions. It's not going to be large in your database, but what you are going to be holding in an entry that corresponds to a specific 11 length word is that you will be. So now imagine you have a table like this. What you convert is you convert these 10 million sequences to a table like this. You have a table, okay, and each row in this table. Now, to simplify these things, let me talk about not 11 base pairs, but just think about if we had 3 base pair length words, okay? Without loss of any generality, we can say that instead of 11, because this is just a parameter class, if we have chosen to use 3 base pair length sequences, you could have 4 to the 3, 64 different such, not 64, such different words. So what are they? So it's going to be AAA, AAC, AAG, AAT, then ACA, ACC, ACG, ACT, and it goes on like this. So in your table, your index would contain 64 rows. Okay, if you use, and here, what we are going to store in this table. So for this row, what are we going to have? Okay, this for AAA. What we are going to store is that which sequences in this table. So we are going to process each of these sequences one by one. And whenever we see an AAA, we are going to store here that this sequence, okay, for example, sequence 1. Sequence 1 contains this AAA starting at the, for example, 10th position. Okay, so this information in this table, what we have is that for all these possible words, if, for example, another sequence, sequence 3, also contains AAA somewhere else. So sequence 3 also contains this at the 15th position, for example. So we are going to have a list of all the sequences which contains this occurrence of this word exactly in their sequence. So what will this give us? It will give us this. If in our query sequence we are going to do the same thing, and if we have an AAA in our query sequence, we will immediately know which other sequences in our database match this query sequence at that position, have exact same match. Okay? So this entry at AAA gives us all the database sequences which exactly have this AAA in their sequences. So this will give us a way to find out a seed region. Okay, this is still not finding this AAA, existence of AAA does not mean anything yet. Okay, you may have, especially if your word size is 11. Actually, 11 is a long word size. We are also going to talk about that. What is the probability that we can get a match of length 11 by chance? Okay, in your database, for example, if you have a matching, this length 11 window exactly matches a number of database sequences. What is the probability of getting this exact match of length 11 randomly by chance? So, especially this AAA, lots of sequences in your database is going to contain AAAs, maybe thousands of them. So, and at that point, the alignment is not done yet. These seed matches are considered as initial seed matches. And after we find that, what we have is something like this. This query sequence in this region matches this database sequence at this region, okay? This is the query, this is the database sequence. And the next step of the alignment, what BLAST does is, it tries to look at these flanking regions at both sides, to the left and to the right. Okay, we have this matching region AAA. Can I extend this matching region to the right and to the left? Does this matching continues? So it maintains the score while extending these matches, these seed matches, and the extension goes on until the score drops below a certain threshold. So it tries to extend in both sides and sees whether we have a significant match there, and after that it stops. The original BLAST, the first BLAST introduced in 1990, did this extension using ungapped alignments. So the first alignments produced by the original BLAST did not contain any gaps. It was just extending it by making use of the Blossom 62 matrix or the DNA scoring matrix. And it was either we had a mismatch or a match or a mismatch. It didn't allow gapped alignment. But the later extensions to BLAST had this gapped alignment. But the whole idea is based on this. Okay? So we have a certain database, index database, which contains the exact occurrences of 11 base pair words in the database. And in our query, by sliding the same 11 base pair like window over the sequence, we can very fast, very quickly find which database sequences contain this exact 11 words. Okay, we don't need to go over all 10 million of them because we have a table entry for it. We can immediately go to that row and see which database sequences have this exact. So this speeds up the search a lot. Okay, so this is the whole idea of BLAST is based on this. And there are certain optional tunings that can be done. For example, in a database sequence or in a query sequence, there may be regions of DNA which does not contain much information. For example, a region which is 200 base pair long and it's all C's, okay? C. So it's, such regions are called low complexity regions. In terms of information theory, these regions may be, and do not forget that the genome, most of the genome has non-coding sequences, non-coding regions, which do not encode a gene. So if you're doing a genome search, there are many regions on your genome which are not meaningful. There's no need to search those regions. So these low complexity regions may be filtered out, may not be used in your searching. And these query words of length 3 or 11 for DNA sequences are created from the query sequence by sliding a window over the sequence. So we have these MEF, EFP, FPG, PGL, GLG subsequences. And what we do is, since we pre-processed our database and created such a table, what we can immediately do is, when I get this MEF, I can search which database sequences contain MEF in them. Okay? It's very fast, I can do this. I don't need to go over all the database sequences and search whether they have MEF. This is already done previously. The search and this table creation, that's the very important point, is done, this indexing is not done on the fly. I mean, you do this indexing once, you store this your index in some place, and for each query, you don't do this indexing from scratch again. Okay, this is done. This is a pre-processing step. This index building time does not count in query time. Query basically uses a built index. Okay, you don't build this index all the time for each query. However, you need to consider this. If your database is dynamic, and which the biological databases are dynamic, meaning that if every day new sequences are found, you should also consider how are you going to update your index. Your indexing algorithm should be able to have this updating mechanism, and it should also be fast. For example, if you have a new sequence in your database, if you need to build your database from scratch all the time, it may take a lot of time. So, but this kind of a strategy, look at this table building strategy. If we have a new sequence, is it going to take too much time? No, right? All we need to do is, okay, this is a new sequence, just go over its words and just update these table entries by adding this new sequence if it contains that word. So the table, having new sequences will just be appending some data to our existing index. Having this new sequence does not violate the information of the previous sequences. So it's just additional information. You don't need to do the whole index building from scratch again. Okay, so these are important factors in database research. How update of the indexes, the size of the index building method. And what we do is, we scan this table after we have found these words. This index preprocessed database is searched for, these are called query words, these words of magnitude or 11. And each match is a seed for an ungapped alignment. This is in original class. So this is after we have found which database sequences have exactly the same word, this becomes a seed match for an ungapped alignment. Okay? And BLAST actually uses high-scoring words created from words similar to query words, meaning that BLAST probably does not use this table idea. Okay? This table idea uses something similar to this. But you need to read the BLAST paper. The BLAST index does not, this has some differences from this table structure. Probably it does not use this table exactly like this. It also allows for searching similar, not exact. For example, if I have MEF, the indexing search mechanism will also going to give me, for example, things like MFF. Okay, similar query words can also be found in the first step. Even if I don't have an exact match, I can find a high-scoring word by finding words similar, not exactly to these query words. And in original BLAST, the seed match is extended to left and right using ungapped alignments. And our extension, this extension to left and right continues as long as the score increases or stays the same. And we call this high-scoring pair, the query and the database pair, sequence pair. We call them a high-scoring pair. And BLAST 2 actually uses a gapped alignment by allowing a certain amount of gaps, okay? And we using it after we do these extensions for all the database sequences, we have a list of hits. Okay, these are our first hits. These hits are then post-processed so that, for example, there may be 10,000 sequences that are extended and aligned. And you have a score threshold, a cutoff score S, for which you keep only the alignments which gives you a score of S or better. Okay, you have a score threshold you do not keep all the alignments, even if the score is greater than 0. If it's below a certain threshold S, which is this S, number S again is an input to the BLAST, is one of the parameters that the BLAST use. You can change those parameters in advanced BLAST options. Okay, this cutoff score can be decreased. If you want to get more results, for example, you can decrease this cutoff, or if you want to do more stringent, more strong similarity search, you can increase this S value. And after that, this score S does not tell us, it tells us to order the hits, but what the main important contribution, one of the most important contributions of BLAST is that it determines a statistical significance measure, which we are going to see today at the second hour, which tells us whether this score could have been obtained by chance, or do we really have a biological similarity there? Okay, so we get a hit to our database. So are these really biologically related sequences, this hit, or could we have obtained the same score simply by chance? Okay, this is a very important question from the biologist's point of view. Okay, because there's an algorithm, there's some computer program running, and it's giving you some results. But the thing is, the first thing in biologist's mind is, can I trust what this program gives me? Okay, can I trust the results of this tool? And this is what the statistical significance is about. It tells you whether you can, how much you can trust in the results. It's going to tell you, okay, this is the result I give to you, and this result, if I had random sequences which are not biologically meaningful at all, you could have got the same score by chance, by this much chance, okay? Or you could have, in a random database, 3 sequences could have obtained the same score or better score to your query sequence, and it tells you something. Okay, so the statistical significance is an important measure. So this is what we are going to see next. And in summary, BLAST, the most important part is we have an indexing mechanism, and we do query the sliding window, the query verbs, our sequences chopped up into overlapping small sequences, and they are searched very fast exactly, and the alignment is done by extending these fast retrieve seed matches. And extension is the most time-consuming step, but here we do not do the extension for all the database sequences. We do the extension only for these table entries which have exact match to our query sequence. Okay? And they're improving BLAST each time. There are different versions of BLAST. There's Mega BLAST, Psi BLAST. The main thing in Psi BLAST is it uses a dynamic scoring matrix, scoring measure. So this Blossom 62 is something global. You say that, okay, the matching of this amino acid with this amino acid has this score. But it's known that from protein families to protein families, these matching may differ. Okay, the certain amino acid substitution rates may differ. It varies from different protein families to other protein families. So and therefore, we cannot use a single static Blossom 62 matrix and hope that it works for all the protein sequence search. So what Psi BLAST does is it gets some initial results, okay, from the database, and by using an alignment of these initial results with each other, it creates a dynamic scoring matrix based on it updates the scoring matrix based on the first initial results. And then it iterates, it does the query again based on the updated matrix, and it does that until certain convergence, so it's more sensitive to find biologically related sequences. And you can go check BLAST website and perform a simple query. So this is the website posted at NCBI. Okay, NCBI BLAST home. Now, what you can do is you can search your query in a specific genome. Okay, for example, you may have a new sequence, and you may want to find out what is the most similar sequence in the human genome to your query sequence. So you can, so it means that you can have different databases. You can search your query on different databases, on human genome database, on mouse database, or on other databases. Or you can also choose, for example, let's do a protein BLAST. Okay, there are different versions. BLAST P is the first version of protein BLAST. PSI-BLAST, this is the extension I was talking about. It uses a dynamic scoring matrix. So let's do BLAST P. Now, so we enter our sequence like an amino acid sequence. Looks like a protein sequence, but there's no need. I think there is need. There's no need? Oh, okay. Let's look at our, let's check out this. So it's because we have 20 amino acids, we need to make sure that we cannot use all the letters of the alphabet. For example, there's no B, I know that, but I thought there was E. Let's see the letter. So to make sure, we need to, we have E. Okay. So A, C, D, E, F, G, H. So up to N. Oh, there's no J. So we don't have D. We don't have J. But we have all the P, Q, R, S, T. So let's P, D, G, H, I, K, J, E. Okay, so let's search this. Probably it's not going to, shouldn't get any hit, okay? This is just not biologically correct sequence. It's not a biologically existing sequence. So here is, we hit blasts. Also, we choose some databases, okay? So there's non-redundant protein sequences. Okay, searching. Our protein contains 29 letters. So it's still they updated the search parameters since we had short protein sequence. They updated search parameters. But let's see if we have, I think we have some results. Wow, yeah. So actually the results are bad, okay? I think this black region indicates that the score is bad. So if we had good alignments, they would have these red, purple, green colors. But still we got some hits, okay? This region, for example, we had a hit with the hypothetical protein. So there are also hypothetical proteins. These are genes that are found, okay? The genes that are identified in the genome, but still their functions are not known. Even there's the encoded protein, it is not known, so they are called hypothetical proteins. But we get a lot of hits, as you can see. And we get a positive score, okay? About 62% of our query were aligned in the first alignment. But let's see our E-value, okay? This E-value indicates that in the database, 0.29 sequences could have obtained the same or better score. So, and this is like a meaningless English sentence, right? How can you say 0.29 sequences have this? It's like, okay, it's like having a football manager saying that, okay, my team today, we are missing 0.5% today. So the counts, when we count things, we usually use integers, right? But here, e-values also count, but it can also have non-integral values. So sometimes it can be understood as a probability, but it's not a probability, okay? It's a count. Basically, it means this. In the database of non-redundant protein sequences, 0.29 sequences could have got the same score by chance. Okay? So this is what it means. And usually people do not think about this meaning like this when the e-value is less than 1. Usually an e-value of 10 to the minus 3, in other words 0.001, is considered a threshold by many biologists to indicate homology. So you should at least have an e-value, and you should at most have an e-value of 0.001 to have some biological meaning between your query and your match database. So immediately we see this 0.29 is a high e-value, higher than 0.001. So probably what we have here is not biologically significant. This is what it should be. Okay, we just made up a protein sequence and did the query, but still there are some, this similarity we got by chance. The similarity we got here, this first, it's one of these 0.29 sequences. Okay, so, and the e-value of, as you can see, when our fourth hit has an e-value of 4.1, it's like out of the database, 4 sequences could have got same or better score completely by chance, with no relation. So this is itself also by chance. This shows that this match is also one of those 4 points. So let's, for example, do a search directly on a known protein. Okay? So we can, if we do the search for a real protein sequence, we can enter the accession number here. So these are identifiers of these protein sequences. And for a database, we can choose several different things. We can use protein data and proteins are proteins with known structures. If you want to find out whether our sequence has a similar sequence with known structure, then we should search it against the PDB database. Or there are patented protein sequences. I didn't know this before. Environmental samples, Swiss protein sequences, reference protein, and this non-redundant protein sequences means that this 10 million, it may be a huge number. And if, for example, out of this 10 million, some proteins may be similar to each other, more than 95% sequence identity. It means that you have a protein, in the database, you have other additional entries which are very similar to that protein. So the database is therefore redundant. Okay? The non-redundant protein sequences means that sequences with high similarities are removed. Only one representative remains in the database that represents these very high similar sequences. And the main purpose is to increase your search speed. And if you have this, if I find the similarity to one of these representative sequences, then I can find the others easily because they're in the same cluster. We can choose also organism names, etc. And we can choose the program. If we do the search for this real protein sequence, the first thing that BLAST does before actually giving the results is that it analyzes these our query sequence and find out this conserved domains. So in addition to the sequence database, there is a smaller database domain database which have certain regions which has in proteins which have special functions are stored in this database, and it gives us that whether, so it's telling us this protein it has a single domain hot D superfamily. Okay, so it is this sequence has similarity to protein sequences in this superfamily. That they have a specific function. I don't know the biological meaning of the hot D keyword, but it's the name of a certain biological function. So as you can see, probably the first sequence is the one we search for. Let's see. Yeah, the first one was the one we searched for. And there are other, as you can see, lots of, in this database there are lots of proteins with unknown functions. We do not know the function of all the proteins. Lots of them are hypothetical proteins. But the fact that this hypothetical protein has other similar sequences in different species means that it has to be this protein should be something, it should be a real protein. It's hypothetical, but the same very high similar sequences are found in other organisms as well. And if you look at the e-values, as you can see, these are really, really small numbers. Okay, 2 times 10 to the minus 103 means that 0.103 zeros. Okay, and then 2. So into 102 zeros and 2. But it's a very, very, very small number. It means that there are a very small number of proteins that could have gotten this score by chance. Okay, and up to all these results, in the first page of the results, all of them are very significant hits. This one is also a significant hit. Okay, there are many, all of these matches. As you can see, we get many matches to our query protein sequence, if it is really a protein sequence. Okay, so let's have a break and let's continue it fast, quickly in the second hour. I'm going to start how do we compute these e-values? So how can we compute an e-value for class? Let's have a 10-minute break.